Hi micro folks, this is Professor Carberry. This is gonna be day one setting up an experiment, seeing if the type of soil that legumes are planted in has any impact on their ability to form root nodules. So in a previous video, you might recall we introduced the nitrogen cycle and we talked about nitrogen fixation and how this cool soil microbe called rhizobium can invade the roots of legumes. And legumes, you guys, you might recall, are like lentils, beans, peas, clover, alfalfa, lupins. So the rhizobium invade the roots of the legumes and triggering the plant to form these cool little anaerobic bumps on the roots called root nodules. And within the root nodules, the rhizobium performs nitrogen fixation. It takes the unusable molecular nitrogen from the atmosphere and converts it into usable ammonia, ammonia that can be used to make proteins and nucleic acids. So when when legumes are growing with their symbiotic rhizobium partner, they should grow faster, stronger, make more food, right, if we were farmers. So I've always been interested, though, it's like if you bring seeds, beans, peas, clover home and put them in your soil, is there a chance that the correct rhizobium partner wouldn't be present? So this is just a real simple experiment, you guys. So what I thought we could do is we could compare legume growth and root nodule formation using 3 different types of soil, if we want to call that. So the first one, you guys, we're going to use is just good old play sand. Just went to the hardware store, got a 50-pound bag of play sand. It's been washed but hasn't been treated otherwise. And then the next treatment, you guys, is going to be commercially prepared potting soil. And there's many different types. Let me see if I can grab this one because I found this really interesting. So this one, you guys, it says that it contains other non-plant food, including microbes. And so in this, they say they have beneficial bacteria, Bacillus subtilis, Bacillus licheniformis, Bacillus amyloliquefaciens, Bacillus vegetarium, and then Streptomyces liticus. Now, Streptomyces, these are the genus Streptomyces. They're famous for making antibiotics, so this is cool. And then, folks, I was very excited because furthermore, they've included some fungal symbiotic partners, and we will be describing these in our fungal unit. These are endomycorrhiza. They actually grow into the roots of the plants, increasing surface area for water absorption and for mineral absorption. And here they've included 3 different endomycorrhiza: Rhizophagus intradices, Rhizophagus claris, and Septoglomus deserticola. Isn't that exciting? So anyway, guys, so that's the commercially prepared potting soil. And then the last soil, guys, that I'm going to use is just good old dirt, dirt from our garden here. And also, I dug up some dirt from the community garden where our fava beans are growing. And you guys might remember in the previous video, we dug up the fava bean roots and we saw lots and lots of root nodules. So I also included dirt from around the roots where I dug up that plant because I'm pretty sure there's going to be at least the rhizobium that can form root nodules with the fava beans. Now I do not know if those same rhizobium can help other legumes. So, and then what we'll do, folks, is we're gonna plant lentils, and we're gonna plant 2 different types of beans: a black bean and a Maya Koba bean. Right? And folks, this is so much fun. Not only are these legumes good to eat, you can plant them and they'll grow. So very often if you go to this store and buy seeds in little packets, they're really expensive. But what I've discovered is you go to the grocery outlet, you can get a pound of lentils or a pound of beans for 99 cents, and you could probably plant, I don't know, an acre of land with these beans and lentils. One thing I do want to check, you guys, and that is there are some plants that have been genetically altered, and I'm not 100% against genetic modification, but my thought was probably maybe what I should be doing is maybe going to, say, a food co-op and buying organic beans and peas and planting those. So we can get into the whole genetic modification later on. So anyway, so great source, you guys, of lentils or beans to plant in your garden. So what we'll do is in one sand, one potting soil, and one dirt, I'll add lentils. And the reason I'm choosing the lentils is I've been planting them in the garden and they germinate like crazy. It's just really endearing. Like within a day or two, they're sprouting, growing like maniacs. And then folks, in the other ones, I'll plant a mixture of the black and mycopa beans, right? And again, what we'll do is we'll just compare maybe like the speed of growth. Maybe we can compare the height, how tall they get. And then what I really want to look at, and we'll let them mature for at least a couple of weeks, is root nodule formation. So do we get the same amount of root nodule formation in sand as we do in regular dirt, right? Maybe it doesn't make a difference. One thought is, you guys, is when they're harvesting the lentils or the beans in the field, they literally are going to be dirty, right? And the dirt could be a good thing because that could contain the good partner, the rhizobium partner, right? So anyway, just a quick experiment. It'll be kind of fun. Any excuse to talk about food or any excuse to grow things, you guys, I'm right there. Okay, so we'll stop this here and we'll follow up every few days.